Bismillahirrahmanirrahim. Class, today we are going to discuss reproduction in Ephedra. In Ephedra, the plant body, as we know, is sporophyte, and in Ephedra both monoecious and dioecious species are found. However, most species are strictly dioecious, means that dioecious species are more than the monoecious species, and in dioecious species, male and female cones, these are developed on the different plants. Due to the superficial resemblance of the reproductive structures of Ephedra to those of the angiosperms, these reproductive structures are sometimes referred to as inflorescences or flowers. Here in this diagram, we can see a dioecious plant, dioecious Ephedra plant, in which the male and female reproductive structures are found. These are the male reproductive structures and these are the female reproductive structures, and the structure of these reproductive structures, male and female, these resemble to the flowers or inflorescences in the angiosperm. So these reproductive structures are also sometimes referred to as the inflorescences or the flowers in the angiosperm plants. In the Ephedra plant, both the male and female cones are compound structures. These are complex structures. The cone axis in both male and female, it bears pairs of bracts, and these bracts they have either microsporangia bearing or ovulate shoots in their axils. Means around the cone axis the pairs of bracts are produced, and in the axils of these pairs of bracts, male reproductive structure or female reproductive structures are produced. Means that either the microsporangia bearing or ovulate bearing shoots are produced. In this diagram, we can see the branches on which the male strobili are produced, male cones are produced. These are the male cones, and in these male cones the pairs of bracts are produced, and in the axils of these bracts the microsporophylls are produced, and on these microsporophylls microsporangia are produced. These are the microsporophylls which are produced in the axils of these bracts, and on these microsporophylls microsporangia are produced. Similarly, this is the branch on which the female Strobilus, female cones are produced, and in the female cones also bracts are produced. This is the structure of the female cone. Bracts are produced, and in the axils of these bracts the ovule bearing shoots or ovule bearing structure is produced. This is also another diagram to show the male Strobulus or male cone. These are the male cones on which the bracts are produced. Bracts are produced in pairs, and in the axils of these bracts the microsporophylls are produced, on which the microsporangia are produced. And the structure of this reproductive body or male Strobulus, it resembles to the flower of the angiosperm, in which the microsporophyll structure resembles to the stamen in the angiospermic plant, in which the filament is present and on the upper side of which the anthers are produced. This is a diagram to show the female Strobulus, female cone in the Ephedra plant. These are the female cones, and these female cones also are formed by the production of the bracts, which are produced in pairs. These are two bracts, these are also two bracts. These two bracts are produced opposite to each other, and these bracts are all produced on opposite to each other, and in the axil of these bracts the ovules are produced. Now this diagram also show the male and female strobili. This is the male Strobulus, male cone, on which the bracts are produced in pairs, and in the axils of these bracts the microsporophylls are produced, and at the terminal end of these microsporophylls microsporangia are produced in groups, and this structure, it resembles the structure of the stamen in the angiosperm flower. This is the female Strobulus. In this also the bracts are produced, and in the axils of these bracts ovules are produced. Now we will see the structure of the male cone. Male cones have rounded apices, means the terminal or the upper parts of the male cones are rounded, and each cone, it consists of main axis upon which a number of pairs of bracts are arranged decussately. Decussately means opposite to each other. In one pair, the two bracts which are present, these are opposite to each other, and then lower pair of the bracts is sterile, mean the in the cone the lowest pair which is present at the base of the cone, this is sterile, on which no microsporangia are produced, while the upper bracts they have microsporangiate shoot in their axils, means in the axils of the upper bracts microsporangiate shoot are produced, and each microsporangiate shoot on which the microsporangia are produced consists of a pair of fused bracteoles, and these bracteoles are also called perianth, and a short axis or column is present which ends in 8 to 12 microsporangia, means microsporangial shoot, it has a pair of fused bracteoles and a short axis or column which ends in 12 to 8 to 12 microsporangia, means 8 to 12 microsporangia are produced on the upper end of the short axis. And this column, it elongates when the pollination occurs, means at the time of pollination, this column on which the microsporangia are present, it elongates, it increases its length is increased, and due to the increase in length of this column, the microsporangia which are present at the terminal end, they become free of the bracteoles and the subtending bracts. Bracts are the scale or leaf-like structures which are present in the on the pedicel, and bracteoles are the structures, leafy structures, reduced small leafy structures which are present at the base of the flower. And when pollination occurs, microsporangia are mature, then the column on which the microsporangia are present, it elongates, and then due to the elongation of the axis or column, then the bracteoles and subtending bracts, these remain away from the microsporangia, and microsporangia, they become free from the bracteoles and the subtending bracts which are present at the base of the uh, uh, microsporangiate shoot bearing bracts and bracteoles. Now next we see the diagram of the male Strobulus, and in this male Strobulus, this is the longitudinal section of the male Strobulus. This is the main central axis on which the bracts are produced. Lower bracts, these are sterile, and then here is the short column at the terminal end of which 8 to 12 microsporangia are produced in group. These are also the microsporangia which are present in the group, and when these microsporangia, these mature, here we can see another diagram of the male Strobulus in which we can see the bracts in pairs in the axils of which the microsporophylls are produced, and at the upper ends of which the anther-like structures, microsporangia, are produced. And when these microsporangiate shoot, it becomes mature at the time of pollination, it elongates and it carries the microsporangia away from these bracts and the bracteoles which are present at the base of the flower, and these microsporangia shoot, it carries away these sporangia and it carries the microsporangia away from the bracts and the bracteoles. This is another diagram showing the longitudinal section of the male Strobulus. This is the cone axis on which the bracts are produced in pairs. Lowest bracts, these are sterile, and then on this axis the fused two bracteoles are produced, and in the axils of these bracteoles microsporangiate shoot is produced. These are the bracteoles or bracteoles, these bear the microsporangiate shoot, and on this microsporangiate shoot, at the upper end of this, microsporangia are produced in group, means 8 to 12 microsporangia are produced. Here also two bracteoles are fused, and in the axil of these bracteoles microsporangiate shoot is produced on which the microsporangia are produced. And at the time of pollination, this microsporangiate shoot, or on the column of the this shoot on which the microsporangia are present in group, this column increases in length, this column increases in length, and these microsporangia, they are carried away from these bracts and the bracteoles. This is the diagram of the male cone. So class, today we have discussed the reproduction in Ephedra and then the structure of the male cones in the Ephedra plant, and next we will discuss how these microsporangia are developed and what is the structure of the microsporangium.